The disease that we're trying to develop new treatments for is called neuroblastoma. It's a childhood tumour. It's a common one. After brain tumour, it's the most common solid tumour. And quite a lot of children who present with neuroblastoma have an aggressive form of this disease. And unfortunately, with the current treatment strategies, which include things like chemotherapy, radiotherapy and surgery, the majority of the children with the aggressive form of disease don't survive. And that's why really clearly new treatments are needed. Our strategy is developing immunotherapy and specifically utilizing T cells for immunotherapy because we know that T cells are very effective in seeking out and destroying tumor cells. But naturally the T cells don't really do this job, so we need to highlight the tumor to the T cells. And for this grant we're using a strategy called BITES, or binding bodies, and those binding bodies have two ends. On one end they bind a T cell, and on the other end they bind a tumor cell. So basically they recruit the T cells to the tumor site, and then the T cells can start to destroy the tumor cells. And with this grant, sort of two main questions we're trying to answer. We're first trying to work out what format, what design of these binding bodies or BITES is the best one. You're trying to mimic what happens during a normal immune response where you have a T cell and a tumor cells interacting with one another. So you can more or less predict what may be a good design, but we have found that you really need to test a bunch of formats to really find the one that's the most potent. So that's one thing we have to do. And the second question we have to answer is how do we fit this in with the other treatments for neuroblastoma? And what we think will happen is these children usually have quite big tumours, so quite bulky disease. And what we envisage will happen is you use, for example, chemotherapy to try to shrink this tumour down till you have a small number of remaining tumour cells back. And then we expect that this kind of treatment will be more effective at mopping up any of the remaining tumour cells and, importantly, stopping the tumour from coming back, which is commonly the problem in neuroblastoma. At the moment we're using antibodies to do more or less what I described, and we really envisage that this will be a step up from there because now with these binding bodies we use the T cells that we know are very effective and also have a long-term effect on trying to prevent relapses. So what we're trying to achieve is curing more children with this aggressive form of neuroblastoma. And I think the other important point is that eventually what we're trying to do is getting away from some of the other more conventional treatments that are very toxic and having very significant late effects. So we want to cure more children with neuroblastoma at less cost of long-term toxicity.